Started. I am terrible at trying to remember to start these recordings sometimes. So if you ever notice that I haven't started it, just like flag me down, let me know, start that recording. And if you haven't noticed, I did already post yesterday's recording on Moodle, so if you wanted to go back and look at it, it is there for you to peruse. All right, but talking about autotrophs making their own energy here. So living organisms, they need energy. All right, I've said this multiple times on separate occasions in this lecture so far. All right, so these photosynthetic species, they're going to capture light energy, storing it in organic molecules such as sugars, used by photosynthetic and non-photosynthetic organisms as their energy source. All right, so plants, they're capturing light energy, creating these sugars. The plants themselves can use those sugars to go through cellular respiration or other cellular processes. But we could eat those plants and capture the sugars ourselves and use plants as our own source of energy. All right, now I mentioned cellular respiration, and we're not really going to get into that in this class, but I wanted to throw it out there because plants also go through cellular respiration. All right, so cellular respiration is the process by which living cells are obtaining energy from organic molecules. All right, so they're using organic molecules such as those sugars, such as glucose molecules, and breaking them down and giving themselves energy. All right, so we go through a lot of cellular respiration ourselves. All right, but we often don't think of plants as going through cellular respiration, but they do. All right, they have cellular processes that they need to go through. They need energy themselves, so they are breaking down the glucose that they have made to provide themselves energy. All right, now their primary aim is to make ATP. All right, so ATP you can think of as like the energy currency of the cell. All right, this is carrying along energy, and this energy can be used in other parts of the cell as needed. All right, most respiration is going to use up oxygen. All right, oxygen is consumed and carbon dioxide is released. All right, so even though we think of plants as releasing oxygen, they also release small amounts of carbon dioxide because they go through cellular respiration. All right, now this will become important when we talked about maybe plants that go through flood stress and don't have access to oxygen anymore. All right, so keep this in mind, but again, I'm not going to make it the primary focus of this class. All right, just to kind of reiterate this cycle. All right, I really want to ingrain it in your minds here that we are using light energy to create these sugars. These sugars are being used in cellular respiration to create carbon dioxide, water, but provide us with energy. And then the cycle goes through once again. All right, so it's this whole cycle that we're going through. All right, so we talked about leaf anatomy before. I just kind of want to reorient yourselves to that leaf anatomy, because now we're going to specifically focus on the part of the leaves that goes through photosynthesis. Right, can anybody remember what type of tissues support photosynthetic cells? All right, what are our three types of tissues? Dermal, vascular, ground. Yes, so which one of those three would contain photosynthetic cells? Ground, yeah. They're not dermal, they're not providing protection, they're not vascular, they're not providing transport of nutrients or water, so they are ground tissues. All right, so to orient ourselves to our image here, we have our leaf vein with our xylem and our phloem. This would be our vascular tissue. All right, we have our dermal tissues. All right, they are at the top here, our upper epidermis, our lower epidermis, any cuticles, guard cells, trichomes. All right, but when we're talking about cells that are photosynthetic, we're talking about our ground tissues. And we can break these ground tissues into some subcategories here. So we can have palisade mesophyll. All right, so those cells are these cells that kind of look like columns towards the top of the leaf. All right, this layer is called palisade mesophyll. They function in photosynthesis. They're often the first cells that will come in contact with the light. All right, below the palisade mesophyll, all right, we have spongy mesophyll. All right, so these are these irregular rounded cells that are on the bottom of the leaf. You can see there's a lot of airspace between those cells there. All right, they also function in photosynthesis, right? But the space between them also increases their surface area for gas exchange. All right, so gas exchange, we're talking about needing CO2 and releasing oxygen. Now there's also these types of cells surrounding our vascular tissue that I just wanted to point out. All right, these would be bundle sheath cells. You might see that in a lot of diagrams here. I'm not too worried about you knowing about bundle sheath cells, but I did want to include it because it's a pretty prominent feature in a lot of these cross sections. All right, these cells basically help regulate what goes in and out of the vascular tissue. All right, so we looked at the leaf. We see we have spongy and palisade mesophyll. If we zoom in on one of those mesophyll cells, all right, we're going to see chloroplasts. And these are the organelles where photosynthesis is occurring. All right, so the green pigment that we see in our chloroplasts is called chlorophyll. All right, that's a pigment that's specifically taking up the light energy. All right, the majority of the photosynthesis is going to occur in those mesophyll cells. All right, carbon dioxide is going to enter, and oxygen is going to exit through the stomata in our dermal tissues. All right, so if we zoom in even further, we see our specific organelle, chloroplast. All right, so if we look at a chloroplast, there's some anatomy that we need to kind of use to orient ourselves here. All right, so in our chloroplast, we actually have two membranes. Right, we have an outer membrane. All right, on the very outside, we have this intermembrane space, and then we have our inner membrane. All right, so chloroplasts, they have two membranes separating them. All right, now we also have a third membrane system, the thylakoid membranes. All right, so a thylakoid is one of these flattened disc shapes inside of our chloroplasts. All right, they're basically essentially a hollow sac that has our plasma membrane surrounding that sac. All right, so there's a lot of membranes within a chloroplast because we have these thylakoids, right, these membrane-bound sacs stacked upon each other in a formation known as a granum. All right, so we have multiple thylakoids stacked upon each other to form these granums. All right, so essentially we have this network of membranes. All right, now inside of our thylakoid, we would call that the thylakoid lumen. Outside of our thylakoids, all the space surrounding our thylakoids would be the stroma. All right, so this terminology is going to become important when we start talking about the details of photosynthesis tomorrow. All right, so big takeaway here, we have a lot of membranes, and it's within these membranes that part of photosynthesis is occurring. All right, so I just want to kind of step back, reorient ourselves, give you a chance to think over what we've talked about so far, and draw a leaf cross-section to the best of your abilities, all right? I'm not looking for artistic content here. Just draw it out so you have some muscle memory. Label the mesophyll. I would label them spongy and palisade, all right? Be nice and specific. The epidermis, the vascular bundle, and the cuticle, all right? Give a brief description of what they do, all right? We're kind of looking back what we did yesterday, all right? And I would also indicate which of those cells is contain chloroplasts. And you can work with each other and collaborate. Get those the Wednesday blues out. Seem very groggy. That would. Yeah.
These are my favorite markers that exist. So I keep them with me everywhere.
So as you're drawing your mesophyll, also try to draw palisade versus spongy mesophyll. Yeah. No. So the epidermis is basically like a one cell layer. Yes. Yep. Low. The cuticle kind of encloses the epidermis. It's a waxy layer very on top. Yes, you have it first. Alright, so it looks like a lot of you are finishing up or have already finished your drawings. So let's come back together and draw one together. Alright, so this is the top of my leaf, this is the bottom of my leaf. Alright, if I were to draw cells, and they are not going to be neat by any means, I am not an artist. Alright, but if I have one layer of cells, top and bottom, what would that be? The epidermis, yes, the top epidermis and the lower epidermis. Now I heard the word cuticle, right? Where would the cuticle go on this image? On top of, above the epidermis. Yes, yes. So it's going to be this waxy layer. It's not really a cell layer, just a layer of wax. That's going to go above them, or if we're talking about the lower epidermis, right, just to the outside here. All right. So let's see. If I were to draw the vascular bundle, I'm actually just going to draw it out here. All right. What might be included in the vascular bundle? What types of cells? Xylem and phloem. Yeah, so we'd see xylem and phloem within our vascular bundle here. I'm not going to attempt to draw those cells. So we have our vascular bundle, very prominent in a lot of images here. What about our mesophyll cells? What type of mesophyll cells am I going to find at the top of the leaf? Spongy or palisade? Palisade, yeah. So there's going to be these very columnar-looking cells here. All right, and I drew them in green for a very specific reason. What do they do? What do they function as?
Harvest light.
They harvest light. Yes, they go through photosynthesis. All right, so that leaves really one space left down here. What type of mesophyll is down here?
Spongy.
Spongy, yes. All right, I'm going to draw it sort of like this. What does spongy mesophyll do? Gas exchange, but also, same thing as palisade. Photosynthesis. They also harvest light. Yes. All right. Did I miss anything? Well, it's hard to tell with the blue screen. Looks like we got everything there. All right, any questions about leaf anatomy? I know it's a bit of a review, but I wanted to get it down. Yes?
Was the palisade cells beyond the top and bottom, or just the top?
Just the top. So, I really didn't mention this, but these palisade mesophills, they actually try to focus the light as the light passes through them, and then it can bounce off these spongy mesophyll cells. All right, so their shape is very much on purpose. All right, any other questions? All right, so we're going to dive a little bit more into photosynthesis tomorrow, all right, but kind of an overview. All right, we have two stages of photosynthesis. We have the light reactions, which, just like it sounds, uses light energy. All right, they produce ATP, NADPH, and oxygen. We'll go over those tomorrow. And then besides the light reactions, we have the Kelvin cycle, which is sometimes called the dark reactions because you don't need light to go through the Kelvin cycle. All right, and if we're looking at where these occur in the chloroplast, right, our light reactions are happening in the thylakoids, right, all those membranes that are stacked up upon each other, whereas the Kelvin cycle, or the dark reactions, are happening in the stroma, right? So that's that space inside of the chloroplast, but outside of the thylakoids. All right, if we're talking about light, we have to understand light. And hopefully those videos kind of give you a good overview of what light is. All right, so light is a type of electromagnetic radiation. All right, it travels as waves, as you saw in those videos. All right, we can measure those waves. We can measure how tall the waves are. We can measure the distance between the crests of the waves, right, and give us short to longer wavelengths. Now, depending on the length of the wavelength, that's going to determine how much energy is carried in those light particles. All right, so those light particles we can think of as very discrete particles, discrete packages of light, and they are called photons. All right, shorter wavelengths tend to have more energy. All right, so if we're talking about shorter wavelengths, wavelengths that have, and that looks more like an actual wave in the ocean, all right, but they're very short compared to something that might be a little bit longer. All right, more energy, less energy, and longer wavelengths. All right, so you learned about the electromagnetic spectrum. I kind of put this image up here again to see that we have wavelengths that can be very short to very, very long. And as we get into shorter wavelengths, our energy is increasing. All right, now when we're talking about photosynthesis, we're just talking about a small portion of the electromagnetic spectrum. We're just talking about visible light. All right, so everything else besides visible light we're kind of ignoring when we're talking about photosynthesis. We're just focusing on visible light. All right, now within visible light, we see that we have longer wavelengths and shorter wavelengths. All right, again, carrying different amounts of energy. All right, so these wavelengths that are carrying these photons, these discrete packages of light, all right, when these photons are come in contact with another object, all right, they can do a couple of different things. They can be absorbed, right? They can be absorbed into the object itself. They can be transmitted, or basically just go right through the object, or they can be reflected, so they can essentially bounce off the object. All right, so if we look at our chloroplast here, we have light coming in, all right? We see a lot of this light is absorbed, all right? The light that is not absorbed can either be transmitted through, right? So it goes right through our chloroplasts, or it can bounce off of our chloroplasts and be reflected. All right. Now in our thylakoids, all right, those stacks of membranes, we have pigments. All right. So a pigment is a molecule that can absorb only a certain wavelength of light. All right. So they're very specific to the type of light that they will absorb. So if we look at our image again, we notice that they're absorbing some light, but some light is being transmitted out. All right. And that light that's being transmitted out or reflected out, that light is green. All right, so they're not absorbing the green wavelengths. They're absorbing every other color in our visible spectrum except for green. All right, and this is why leaves are green. Leaves are green because the green color is not absorbed. It's reflected back. All those other colors are essentially captured within our leaves. All right. So we do have to talk a little bit of chemistry to kind of understand how photosynthesis and how light reacts with photosynthesis here. So I'm assuming everybody kind of knows what an atom is from high school. You've talked about an atom at some point in your lifetime, I'm sure. But the atoms are the smallest functional unit of life. All right. They can be composed of subatomic particles. All right. Those are your electrons, your protons, your neutrons, all right? And if we take a bunch of atoms and combine them together, that is how we form a molecule, all right? So on our image here, this is a carbon atom, all right? We have our protons and our neutrons in the middle, and we have our electrons orbiting this middle nucleus, all right? We have our electrons on the outside. Now when we're talking about light energy and how to harvest light energy, all right, we're really going to be focusing on the movement of these electrons, all right? So these subatomic particles that are orbiting around the center of our atom. All right, so when light comes in, right, light is coming in, it's making contact with an atom, all right? It's being absorbed, and it's going to boost this electron to a higher energy level. All right, so notice our electron in what's called the ground state is orbiting pretty close to the nucleus. All right, when light comes in, when our photon comes in and excites this electron is going to move into an orbital that's a little further away from our nucleus. Right, so now it's orbiting a little further away. All right, when it moves into this outer orbital here, we call that an electron in the excited state. All right, so basically it has absorbed the photon and it has become really excited to the point where it's actually going to move a little bit further away from our nucleus. All right, so when our electron is further away from our nucleus, when it's in this excited state, all right, it's very unstable. All right, it doesn't like to be in this excited state. It really wants to go back to where it was originally, back to that orbit that's closer to the nucleus. All right, so it's excited, it's unstable, it has to release this energy in order to go back down to its original orbit. Now I can release that energy in a couple of different ways. All right, one is in heat. All right, so put an image up here of a very hot day. You're driving on the highway. You might see some heat coming off of that black top there. All right, it can release that energy as heat to get back down to that ground state. It can release that energy as light. All right, so I threw up an image of these jellyfish here. They are exhibiting biofluorescence here. All right, so they are releasing light as the energy. All right, that energy is being released as light. All right, now these excited electrons, they can also be transferred to other molecules. Right, so instead of going back down to its original orbital, these electrons can move to another molecule and be captured. All right, and this is what is happening in photosynthesis. All right, we are moving these electrons from one atom to another. All right, we're capturing these electrons in another molecule. All right, does that make sense so far? All right, sounds good. So remember when we talked about the electromagnetic spectrum? All right, we have different types of wavelengths. Some are shorter, some are longer, and they contain different amounts of energy. All right, so when a photon strikes a pigment, all right, that energy is going to be transferred to an electron. All right, so that electron is going to become excited, and it's going to raise it to a higher energy state. All right, so that electron is moving away from that nucleus a little more. But depending on what color is being absorbed in that pigment, that electron is going to absorb different amounts of energy. All right, so for example, our red wavelength, all right, has a little bit of a longer wavelength to it, right, has less energy associated with it. So when it strikes our electron, all right, our electron is moving from our ground state to an excited state, all right, but not as much as when a blue wavelength hits it. Right? It's moving that electron to an even higher energy state because blue has a shorter wavelength and contains more energy. All right, so not all light is equal. All right, it contains different amounts of energy, which moves that electron further and further away from the middle of the atom. All right, so we talked about photons intercepting these electrons, that energy being captured. Where does this occur? All right, we talked about pigments that are molecules that absorb certain wavelengths of light. All right, one of the most common pigments in our plants are going to be chlorophyll, specifically chlorophyll A and chlorophyll B. All right, so there's two different types of chlorophyll. They're just differing in their molecular makeup in one area. All right, so notice one has carbon and 3 hydrogen. Chlorophyll B has a carbon, hydrogen, and oxygen. All right, that's the only difference between the two. All right, they're both taking in green light, and they're absorbing this light in the part of the molecule called the head. All right, so in this part of the molecule, this is where the light is coming in, being absorbed, and then this the excited electron can be transferred to another atom, to another molecule. All right, and so these pigments are going to be found in kind of these clusters. All right, so every single one of these little heads with the tails is a chlorophyll molecule. They're all clustered together. When light comes in, right, it's exciting an electron, and that electron can move from molecule to molecule to molecule before being captured by a very specific molecule. All right, so these pigments, they're the ones that where the electron's being excited, and then where that electron can bounce between before being captured. All right, so I mentioned that a couple of different things can happen when an electron becomes excited. Right? We can release that energy as heat. We can also release that energy as light. All right? About 2% of the excited electrons are going to be producing fluorescence, all right, which is light. And I have this video that NASA put out that I think is pretty interesting, and it kind of relates to our astrobotany theme because we're looking at fluorescence from space.
Light gives plants the energy they need to grow. But what happens if plants receive too much light? The answer is they glow. This neon glow is actually happening around us all the time, the result of a cellular process where light from the sun is transformed and released as fluorescent light. A journey into the plant cell reveals how it works. This is a chloroplast, the energy-producing organ found inside the cells of plants. When sunlight strikes a plant, disc-like structures within the chloroplast absorb the light and convert it into energy. However, a small fraction of this light, about 1%, is emitted as fluorescent light. The light exits the plant cell and is released into the atmosphere. The amount of light released can vary depending on factors like the time of day, time of year, and how much sunlight is being absorbed.
Humans can't see this light. Our eyes just aren't that sensitive. To see the fluorescent light that's emitted from plants all over the world, we have to use scientific instruments that are placed on satellites.
Earth observation satellites, outfitted with special sensors, are able to detect this light from space. After multiple orbits, scientists can construct a detailed view of the data. This is what plant fluorescence looks like on a global scale. The visualization was created from 5 years of satellite measurements, analyzed by a team of researchers led by scientists at NASA's Goddard Space Flight Center. Each point of light on this globe represents fluorescent light exiting a plant cell. The greater the intensity of light, the brighter the color. By observing changes in intensity over time, scientists can distinguish stressed, dead, or dormant plants from healthy and growing vegetation.
Fluorescence measurements like these are important because they can be used to develop improved vegetation models. And this will lead to better predictions of how plants will interact with the Earth's environment in a changing climate.
All right, so I think that was a great video to show that even though most of the light that's captured in plant cells ends up in photosynthesis, a small portion of it, right, is reflected back as light itself, and that can be important. Yeah.
So why does it reflect back, or why does a small amount of it reflect back?
That's a good question, and I think it has to do with how efficient the plant is with its light. So this process isn't 100% efficient. There's some room in there that it's just not going to be absorbed. That can be reflected out, especially when a plant receives a lot of light, almost too much light that it can't do all the photosynthesis with it. That light has to go back somewhere, and that can be fluorescence out. All right, good question. All right, any other questions regarding fluorescence? I think that video is kind of cool because you can kind of see the seasons happening in those images as the light becomes more prominent during the summer and then kind of fades away when everything dies back in the winter.
I have a question just out of curiosity about like the types of chlorophyll. Are there like specific plants that have the specific types of chlorophyll? Is there like a difference, or do all plants have both?
All plants have both. And I'd have to look up the answer to that one to see if certain plants might have a higher percentage of one versus the other. That's a very good question. Let me do some research on that because I'm curious. Yeah. Any other questions? All right. So let's go back to my presentation. All right, so we talked about fluorescence, but we know most of that energy is going to be converted into sugars in the end here via photosynthesis. All right, so one of the big things that how this happens is resonance energy transfer. All right, so kind of mentioned this before. All right, so this is where a photon comes in, hits a chlorophyll molecule. All right, that electron becomes excited, and then that electron is going to be transferred between different chlorophyll molecules until it reaches a very specific molecule. And this specific molecule is called the reaction center. All right? It's in this area, all right, that electron can finally be captured and that can help go through photosynthesis. All right? So our electron's bouncing around until it hits that reaction center where it's finally captured. All right? So when we look at resonance energy transfer, we're looking at these complexes, all right, of multiple pigments, all right? And this would be called an antenna complex. All right? That's a specific terminology that refers to this grouping of pigments. All right, now we don't just have chlorophyll. We have other pigments in the leaves as well. All right, they're called accessory pigments. They don't necessarily function for photosynthesis. They have other roles within the plant. All right, they could give their energy to the chlorophyll, and then that energy can go through photosynthesis. Right, they can extend the range of wavelengths that we can harvest light from. All right, so we can get even more energy from other wavelengths. They can also function in protection. All right, so one of the main ones that we see every fall are carotenoids. All right, so in the fall, the chlorophyll pigments are broken down, they're shuffled back into the tree itself and recycled, and what's left behind are these carotenoid pigments. And we see these pigments because these pigments tend to be this orange, red, yellowish color. All right, and I have another quick video that I'll show on different accessory pigments. All right, because I think that's way more interesting than me standing up here talking. But if you are interested, one thing that's very rare in nature is the color blue. All right, it's a kind of a weird thing that happens in nature. And I have this Hank Green video down here on the color blue. It's about a half hour long, which is why I am not showing it in class. But if you're interested, it's up there.
Why we use Miro? Well, when I draw on a whiteboard, nobody sees it.
And we're going to have to listen to a couple ads
in real
first.
time from literally.
He knows a lot about the science stuff. Professor Gene explains. We just learned about plant hormones, which means we are beginning to discuss specific chemicals that carry out important functions in plants. So let's look at some other plant chemicals called pigments. First, we should know what a pigment is. Pigments are organic compounds that give living things their color and may also serve some other sort of function. In plants, pigments give the leaves, flowers, and fruits their colors, but they're also responsible for things like food production. Let's go through the different types of plant pigments now. The first and most important plant pigment you will hear about is chlorophyll. There are actually 3 types of chlorophyll that may appear in organisms which are capable of photosynthesis, those being chlorophyll A, B, and C. Chlorophylls are large molecules with a cyclic portion that binds to a metal ion. This is extremely similar to another cyclic biomolecule found in our bloodstream called hemoglobin, which we learned about in the Anatomy and Physiology series, except that where the heme in hemoglobin involves iron, chlorophyll contains a magnesium ion instead. Chlorophyll pigments reflect green light, which makes plants look green, and they absorb red, yellow, and blue light. The light energy absorbed by chlorophyll pigments excites electrons in the porphyrin ring, which provides the energy required to initiate the process of photosynthesis. Once again, we covered this process in detail in the biochemistry series, so we won't talk about it here, but this is the single most important process to understand regarding plant physiology, so be sure to visit this tutorial if it is unfamiliar to you. Now, to be clear, chlorophyll doesn't facilitate photosynthesis all on its own. Another set of pigments, called carotenoids, assist with the function of photosynthesis. You might notice that carotenoid sounds a bit like a certain orange vegetable, and you'd be correct to associate the two. Carotenoid pigments give plant parts red, yellow, or orange coloration. But beyond this, carotenoids are known as accessory pigments because they assist chlorophyll in the collection and absorption of light energy. Beyond red and orange vegetables like peppers and carrots, carotenoid pigments are also responsible for the beautiful colors you see in autumn leaves, once all of the chlorophyll has degraded. The next group of plant pigments we will discuss is called flavonoids. These are usually found in the vacuoles of plant cells. As we recall, the vacuole is a large water-filled compartment within each plant cell, which which contains organic and inorganic substances that are important for the cell. The most common flavonoids are the anthocyanins, which often produce bright red coloration in plants. Anthocyanin pigments are usually found in the petals of flowers and in the flesh of fruits, but they can also color the stems of plants. Trees with especially vibrant red autumn leaves are expressing anthocyanins as well. The last important plant pigment is a little different from the others that we've covered. Phytochrome is a plant pigment that isn't visible unless we separate it from the rest of the plant. This pigment is less about creating visible color in a plant and more about triggering certain events in a plant's growth and development. Phytochrome absorbs light just like all of the other pigments, but in this case, the light absorbed acts like the flip of a switch to start a new process, such as seed germination, stem and leaf growth, or bud flowering. So with that, we know how pigment molecules give plants their colors, as well as what processes these pigments facilitate inside a plant. But what are all of the different kinds of plants? With sufficient foundational knowledge covered, it's time to start looking at the wide variety of plants that exist on Earth.
All right, I'm going to stop it there. So I think that gave a good overview of other pigments that can be found in plants as well. They did mention that there is chlorophyll A, B, and C. I did not mention C because it's just the least common type of chlorophyll. And again, I'm not sure why, and I'll have to look that up. All right, so the last thing I wanted to mention for this part of our lecture here is that you might see different types of graphs representing light absorbed by plants. All right, one is called the absorption spectra. All right, and this is going to show all the different wavelengths absorbed by different pigments. All right, so we see the wavelengths absorbed by chlorophyll B. All right, we have the relative absorption on this axis here, so up towards the top, that absorption is really high at the about blue wavelength here. All right, so different types of wavelengths are along the bottom. All right, so we can follow the absorption of these wavelengths for different pigments. We can also look at, as a whole, all those pigments combined would be the action spectrum. All right, so instead of separating out every single pigment and the light that they absorb, right, we're just looking at it as all combined as a whole, the rate of photosynthesis. All right, I'm going to say let's take a 15-minute break here. When we come back, we'll start a class activity that is actually up on Moodle, and I will make sure it did indeed post. But take a break, do some jumping jacks, stay awake, and we'll resume class at, let's see here, 11:25, 11:40. Doesn't look like we got very refreshed on our break. Ooh, all right. Well, you still got 2 more minutes if you want to do jumping jacks or anything. That chair is really in a bad spot. All right, well, it looks like everyone's back. So I think instead of immediately doing the class activity, which if you started on it, awesome, great. Let me finish up the last few slides I have for you, and then we will move on to that activity together here. So that class activity I did put on Moodle. Right there if you want to download it and be able to type on it yourself. That's awesome. But before we move on, let's just finish up the rest of this lecture here. All right, so we've talked about visible light, what happens to pigments when visible light hits it, all right? But we know there's a lot of other light within our electromagnetic spectrum. Fortunately, we're shielded from a lot of it here with our ozone layer on Earth. But what happens when plants might be exposed to light outside of the visible spectrum? All right, well, there's really not a lot of good research on this, I've noticed. Every research article that I found, the experiment that they set up wasn't the best experiment. All right, but they've kind of hypothesized a few things that could happen. So with UV light, they've guessed that it's going to lead to reduced growth, reduced photosynthesis, reduced biomass, which is just all the mass that the plant has, the actual weight of the plant. So if it's growing more, it's going to add more weight, more biomass. Gamma rays, all right, they can actually penetrate into cells, all right. If they're exposed to these gamma rays, it might accelerate the ripening of their fruit, breaking down the cell wall. It could influence development or even function. And if we're talking about maybe some longer wavelengths, like radio and microwaves, it could lead to less healthy plants. And this is specifically the wavelengths that I found the least amount of research on. For example, one of the articles looked at these 2 different trees in a city, right? So just 2 trees, not even the same species of tree. Right? And they noticed one was next to this transmitter and it looked a little less healthy. All right, so it's not the most experimentally sound research out there, but it could give us an idea of maybe something could be there with these radio and microwaves. All right, there are other influences of light on plants besides photosynthesis. All right, light is crucial for making sugars, but also for germination. All right, some plants actually need a light cue in order for their seeds to germinate. Flowering, so day length can indicate when they need to flower, as well as growth. All right, if we're photosynthesizing, we're producing sugars. If we have enough light to do so, we're going to grow even more. All right, so if we look at our response to light, all right, it's an important stimuli. We know it aids in photosynthesis. It helps the plant detect passages of time, specifically with photomorphogenesis. All right, so that term is just engulfing everything plant growth and development that is triggered by light. Right? So it's pretty much an all-encompassing term for a plant's response to light. All right, one of the pigments that we learned about from Professor Dave was phytochromes. All right, so phytochromes can sense different light, right? Specifically red and far red light that signal the seeds to either germinate or not, and can signal the plant to either grow tall or grow bushy. All right, so if we look at red light, if a plant receives more red light, right, it's going to germinate. Right, so if we look at our image down here, we have a petri dish full of seeds. We can see that they have germinated under red light, right, and less so under far red light. All right, there's still some germination, but it's less. Versus if they're in the dark, I only see a couple of seeds that germinated. All right, so red light tells seeds it's time to germinate. All right, red light can also lead to bushy growth. All right, so it's typically the first light that is absorbed by the plant is going to be red light. All right, more far red light will be transmitted through and absorbed by other cells. All right, so if they have plenty of light, they don't need to grow to find any more light, so it's going to lead to bushier growth. So if we look at our plants down here, all right, we have high red light to a little less far red light. All right, we have bushier growth. All right, we don't need to search for sunlight. We can grow nice and bushy. Versus if we have plants that maybe have grown in the shade, all right, the red light has been picked up by the plants above them, which only leaves them with far red light, right? They're going to grow a little bit more taller, right? Because they need to seek out some more sun, seek out better light. So this cues the plant to grow tall. All right, light can also can influence photoperiodism. All right, so that's a physiological response to specific night and day lengths. All right, so plants can sense when it's night and when it's day, right, based on the light that they're receiving. Specifically, when they flower, can be controlled by night and day length. All right, a lot of greenhouses can manipulate light and day length and get flowers that might not flower at that time of year to produce flowers when they normally wouldn't. All right, so plants detect these periods of light called photoperiods. All right, so depending on the plant, they're going to flower in response to different photoperiods. So if they're a short day plant, they are going to flower when the day length is shorter than a critical number of hours. All right, so if we look at this graph here, this dotted line, that is the critical number of hours. And now I'm not specifying a specific number of hours there because it's going to vary depending on the plant we're looking at. Right? For some plants, it might be longer or shorter than others. But it's this critical period, right? This critical number of hours. So when the day length is shorter than that, all right, so if we look at our middle flower here in the first image, the day length is shorter than that critical number of hours, right? That signals the plant to flower, right? Now the opposite is true when we talk about long day plants, right? This is where the day length has to be longer than that critical number of hours. So if we look at our first plant on the second image here, we see that the day length is longer than that critical number of hours that it needs, right? That's signaling the plant that it is time to flower. All right, now often this correlates with the time of year, right? We get more light in the summer, less light as we transition into fall. Depending on the plant, they're going to pick up those photoperiods, and that's going to determine when it's going to flower, right? Hence why we get flowers in spring. We also get flowers in fall from different plants. Now we also have day neutral plants, right? They're unaffected by day length, right? The day length does not influence when they flower. All right, now one thing that they found, right, we've been talking a lot about day length here. It was actually found that the night length was a little bit more critical. If we interrupted the night length, all right, that would determine when a plant would flower. So if we look at this long day plant down at the bottom, they introduced a flash of light in the middle of the night, all right, that basically broke the night into two smaller sections, all right, determining that it was a short night plant. All right, so nighttime actually is more important than daytime. All right, one of the other things that we all know at this point is phototropism, right? Plants' orientation in response to a light source. All right, we can have negative phototropism where we grow away from the light, positive phototropism where we grow towards the light. Typically, shoots, leaves, and stems are going to be positively phototrophic, whereas roots are going to be negatively phototrophic. All right, but how does this actually work? All right, so this is the part of phototropism that we have not gone over yet, and it has to do with this hormone called auxin. All right, so auxin is a hormone that causes cell elongation and can help regulate our growth in a plant. All right, and it's produced at the very tip of a plant. All right, so if we walk through an experiment that was done up on the very top here, all right, we see light is coming in from the side. If we do nothing to our plant, that plant is going to bend towards the light. It is exhibiting positive phototropism. Now, if we cut off the tip of the plant, all right, that plant is going to grow straight up. It's not going to grow towards the light. Right? This indicates that something in the tip of the plant that is causing this bending movement. All right, now that we know that it's something to do with the tip of the plant, all right, these researchers took it a little further and they put different types of caps on the plant. Right? One was opaque, right? So no light can penetrate through that cap. No light can hit the tip of the plant. And one was clear. Now the clear cap, light was able to penetrate through, and this plant exhibited positive phototropism. All right? Just to be sure that it was definitely the tip of the plant and not any other part of the plant, they also repeated this with an opaque shield towards the base of the plant. And they saw that it also exhibited positive phototropism. Right? So this reinforced the idea that there was something in the tip of the plant that was going on. One thing else that they did, a different set of researchers here in 1913, all right, they actually separated the tip, replaced a piece of gelatin block in between the tip and the rest of the plant. Right? And this gelatin block allows the tip of the plant to still communicate with the rest of the plant. Right? There's still communication happening. So when communication was still happening, we saw positive phototropism. Right? But if we cut off the tip of the plant and replaced it by this piece of mica, which does not allow for communication, right, we saw that once again it grew straight. All right, so the tip of the plant has this hormone called auxin. All right, so it was later discovered that auxin is playing the biggest role in causing phototropism. So essentially, when light is hitting the plant from an angle, all right, it causes auxin to collect on the shadier side of the plant. Now, auxin causes cell elongation, right? So it's going to cause these cells on the shadier side of the plant to elongate, and that basically helps bend the tip of the plant towards its light source. All right, so auxin plays a huge role in phototropism. Quick side note is that auxin plays many different roles in a plant. This is just one of the important ones. All right, I'm going to leave this here as a couple links that you will need when you do your class activity here. But at this point, all right, I'm just going to introduce the homework. I have a podcast for you to listen to, a video for you to watch. That's what the quiz will be on tomorrow, as well as the same thing as before, exam 1 on Monday and endurance on Tuesday. All right, but take the next, 15-20 minutes and do the in-class activity on light with a partner here. There is a portion where you will need to draw, and you can draw on a separate sheet of paper. That's quite fine. You're not going to turn this in. This is just to help try to find any problem areas within our lecture today that I can help explain. All right, once we're done with that, we'll move on to talking about our final project. One thing is the final project, you will be working in groups of 3. So if you also wanted to use this time to try to find your group members, it would be a good use of time. Yeah, it'll be like multiple choice, short answers, false, read or like fill in the blank type of questions. You look at the study guide that I posted on Moodle. I have talked a little bit about the format.
We're not much.
Yeah, what do they consist of? Membrane, yeah, membrane with a space. Awesome. Two kinds of plastic cell. Yes, yes. Inner, outer,
That's all I need. Yeah. The membrane.
yeah, space on the inside, but outside of this.
Yeah. And then the full one is called, I colored it up, but it's a gray. So like each disc has like. And then this stuff. I know. I love that you have the markers. It's so nice. And then this. Wait. Oh, so this is like the space. Yeah. So absorb certain colors. All this like that light has all of these. Yeah. Just like the extra space in the inside of it. Yeah. Yeah. What the that's yeah. Oh, chloroplast. Yeah. It's a yeah. So I already answered. Okay, fair enough. So they need to add this to the color, right? Everything is like, so like every color is every other color is like the one that looks like, yeah, colors align. Yeah, it reflects the screen. Wait, I can look at what I wrote down, but I'm pretty sure that. Like the shorter the wavelength, the more photosynthetic. Oh, okay. So. Okay. Oh wait, you said. Well, red I think is one of the yeah, that's right.
And red light is the slowest, the shortest possible wave. Like, which would make like sense.
You can have different shades of red. Yeah, because you can have different shades of red. So see how it affects the sleep. Because the different shades of red are different. So it's larger.
Say
Oh, yeah. Is that where you're from? Or?
no, my brother lives there.
Oh,
Yeah.
that sounds fun.
They love them.
Yeah.
So we're doing the one about the red algae pigments and stuff. I was scared about the question, like, so like, because one is like how to measure this, so like, the different, like the shorter the wavelength, or affects like the color, right? Like the shade of it.
Yeah, yeah. So think about that electromagnetic spectrum. Yep.
Right, so then would test it experimentally, could you use different shades of red? Or like,
You want the right
I don't know why it's
Yeah. You could use. Well, first of all, let's talk about the first part. So what wavelength is this?
Yeah.
Focusing
It's
in?
like red's on the other end.
Come on.
It's most when the wavelength is shorter.
Because that one has them.
Yes, but it can absorb a range of.
It absorbs.
Well, it's-
Why is that?
Yeah, I know, but like strawberry.
Oh.
Not using it,
But this
Has
bar.
the, okay, and the algae is red.
but the algae is red. Where's
Let's see what it does.
colors?
Yeah.
Red. Yeah. So that means all the other colors are being what? Absorbed. Yeah. So it's being absorbed.
Oh, okay.
Oh, no, it gives us.
You say that they bring you up. Okay,
Now you are correct in
yeah.
thinking that this is what the wavelength of more it's gonna be.
Yeah.
Oh,
But it's just not using red because the plant is red.
Exactly. The colors are reflecting it back. Yes. So that leads us to how to measure this experimentally. And this is meant to kind of, it's a harder question. You really have to think about it.
So could you use different colors on the algae?
Use different color lights.
Yeah.
Yeah, yeah, you could. Yeah, so if you use different colored lights, basically you can shine different light from the measure the amount of oxygen that's involved.
How do you
More oxygen. There's actually a really cool machine that we have here at Neighbors. Yeah.
Really?
You actually clamp something onto a beam.
And you can measure how much oxygen.
Yeah, it measures the air content inside of the chamber, and it can measure the increase of oxygen.
Does something flashbacks to the pigment lab?
Yes, yes.
Okay, well I guess that's our answer.
Yeah, so that's one way you can measure it.
On the algae.
So with a machine.
You could measure it with a machine, measuring different types of light. Yeah, yeah.
You're just launched into the sun. And there they go.
Yeah.
They're close enough to the sun at that point. Just tie a rope on one end, pull it back.
Yeah. All right.
Yeah.
So let's think about the
They
first
fried
question.
plants.
One wavelength.
Crispy flavor.
if we think about what makes a leaf green, it's green because it's not more.
Yeah, I could also measure
This might be red. Red.
Oh,
Basically,
yeah.
the wavelength of the size of red.
Yeah, that's true. Like,
Blue green.
or honestly, I already did these.
So
There was like a short
to
place.
measure
Oh, rays. To measure how well the if it's more, if it's less powerful, it's gonna inhibit the cells. If it's a more powerful, light on the spectrum, then it's going to make it like it could make it grow faster. It could penetrate the cell walls and make it do all sorts of crazy stuff.
I feel like for like
Right. So yeah. So like let's do it.
and we mentioned one more.
Yeah, because that's what
I know around this is like my
Oh no.
Oh, this is. Oh yeah, ultraviolet on one side. Okay, so then this one I guess that's harder.
Yeah.
Why don't we
Yeah.
So when you're answering that last question, remember there's a couple of links in the PowerPoint slide to articles that will help you answer that.
Literally one that says why not use something.
Okay, perfect.
Yeah.
Well, we know where we're going.
Sorry, answer yourself. I like it.
That was I didn't know what the answer was, but I didn't expect it to be like what? In no long periods of darkness, the isotopes receives daylight for 45 hours. So they're also
Are those links not showing up?
Oh, now they.
wait, so
Okay, good.
partly because like you know they're
So that's one of the videos
have
we
a
watched.
person, so
It's like the last slide.
is kind of not.
Oh.
Okay, and then flexibility. So in 2017, NASA designed
Yeah,
the second
activity.
much more difficult.
So yeah, it's the one right before the homework, and you don't see any links.
It's just like they didn't want to deal with it.
I can put them up on Moodle if they're showing up. All right.
Why not use all those windows?
Little like chambers.
Yeah. Oh, no.
Yeah,
and then you like get it.
and because of their it would also and then it would like fresh rather than
I don't even know how that all works.
so what there's other stuff like other stuff about the plants.
Yeah, around the plants.
Do you have more questions? No.
Oh, nice.
Although to be fun, I could take a picture of
I hope you can read it okay. If you don't want this, because you have a picture, I will gladly keep it.
it'll be in my signed in forever in my phone. Oh yeah. Always facing this way, the sun.
All right, I'll give us a couple more minutes just to finish up those last two questions. In the meantime, if you want to start looking at the final project document, feel free. We'll talk about it together here.
There you go. Oh gosh, it's gonna. I was just ranting about that yesterday. Because I'm not. Yeah, you were. Holy shit. I have to find like the specific episodes that work for this. Okay, I just. Okay, because I know it's on. But I bet I could find like a few. Just take a look.
I know Macy was sitting next to you. Do you know Macy?
Yeah, we're both from Brainerd.
Oh, okay, got it. So, wait, so then you have her working with at you two if you're deciding to be a group.
Yeah.
She just had to self-isolate out of caution for the last couple of days.
Yeah, I can message her.
All right, that'd be awesome.
So there's this one.
Yeah.
That we can maybe use.
All right, so let's come back together. Let's think about that last question. All right, why don't we use sunlight to grow plants on the International Space Station? What are some of those reasons?
Because windows are annoying to make.
Windows are annoying to make. Yes, it's something you might not think of. Totally. What's another reason?
The cycles of day and night are much shorter, so it messes up the plant's circadian rhythm.
Yeah, those cycles of day and night, it's gonna mess the plants up. Plants have a circadian rhythm, just like us. What's another reasoning?
Using just like lighting technology is like a lot more consistent, so it's like not as much of a hassle.
Yeah, it's more consistent, it's more efficient. Definitely. Is there any other reasons that we thought of? That we saw on the layout? I think that was one of the main ones. One of the other ones I had jotted down is just that it gives us a little bit more flexibility, not having to rely on when the sun's going to appear. Awesome. All right, so let's turn our attention to the final project. All right, so this project you'll be working on throughout the rest of January here. And I'm going to say working in groups of 2 to 3. It looks like we're maybe clumping more naturally as groups of 2, which is okay. As long as we find enough resources for everybody here. But our final project is looking at astrobotany in pop culture. All right, so I know a lot of you have probably seen The Martian. All right, and that's one movie that I'm going to say that is the only pop culture one I am not going to allow because I will be using that as an example a lot in class, which would give you a very unfair advantage. All right, but like a movie like The Martian, right? We are seeing plants grown on Mars. How realistic is that? Right? And that's kind of the question you're going to be asking about your pop culture piece. How realistic would it be to grow a plant in Mars? Now, in the case of The Martian, we actually know that the way he did it was more or less pretty realistic in how he went about doing it, right? But there's a lot of cases out there of pop culture having plants growing during space flight, providing us oxygen during space flight, trying to terraform a planet with plants, all right? So I want you to find a piece of pop culture that has plants growing either on the moon, on Mars, or in a spaceship, all right, during space flight. All right, and I'm going to limit us to those 3 areas because we know the conditions of Mars, we know the conditions of the moon, and I'm going to assume the conditions of space flight for every type of spacecraft out there is going to be more or less similar to what we have now. All right, so it's your task to try to find some ideas for pop culture pieces that you want to work with. All right, so I'm saying pop culture because I'm not going to limit you to just a book or a movie. All right, it could be a comic book, it could be a board game, it could be a lengthy comic strip. All right, anything pop culture related, a TV show perhaps, maybe a specific episode. All right, plants don't have to be the main focus. All right, so I know in The Martian, plants were more or less the main focus. But maybe you are reading a book about someone created a park in a spaceship. All right, maybe that's something you want to focus on. Even if it's not the main focus of the book or the piece, you could still focus on that aspect. All right, so I'm introducing it now because it's going to take a while to probably brainstorm some ideas. And that's one of the things I want you to do today and turn in by the end of the day. Not necessarily the end of the class, but some ideas for your brainstorm. And I put a submission. Did I put a submission? Yes, I did. Submission here. And I do have a document there that you can use as kind of a guideline to how I want you to submit it. Basically, I just want you to come up with at least 5 ideas. All right. Then give me the idea, the year that it was made, the type of pop culture piece. Is it a book? Is it a movie? Is it a comic? And its location. Where is it taking place? All right, now I put spaces on there for 10, all right, because it looks like we're going to have maybe up to 12 groups here, probably 10 to 12 groups. So you might not be able to get your first choice. I'm going to do my best to appease everybody here, but I know there are some pieces that are more popular than others. So with that, I'm going to let you form your group and start brainstorming for the rest of this class and ask me questions. All right. For the final project, it is 45 points. And I totally got rid of it here. But it's broken down into different assignments. So it's not one assignment that is 45 points. All right. It will be multiple assignments that will add up to 45 points. All right. Your final paper is going to be the bulk of that. You will also have to do a presentation. All right. The presentation has to be visual, but I'm not going to limit you to doing a PowerPoint. Right? It can be any type of visual, creative presentation you can think of. All right. I'm also going to ask that you do a newspaper review, kind of like you would see a critic's review of a play or something on in a newspaper. That way, the goal of that, I'm hoping, is to maybe create a poster that we can put in the hallway up here with kind of our review of our astrobotany pop culture. All right. So I'll let you get started on here. You can take the rest of the hour to do so. If you feel confident, I'm not going to make you stay and do it for the rest of the hour, but it would be a great chance to ask me what is valid and what isn't valid to use. All right, so go ahead and start collaborating.